Hello, I'm Sang Lee from Gyeongsang National University in Jinju, Korea. So I'd like to thank the organizers for inviting me to this wonderful workshop with a set of the most active network scientists in the world. And I'm really happy to present my work on the scale-dependent community reorganization of networks from modularity maximization. So there are many precious collaborators of mine and who are actually roughly color-coded according to the topics. I especially thank my postdoc, Won-Hee Jung here, sitting over there, who has been leading this ongoing research topic as the actual investigator or AI, along with my community collaborators. So first, I will give you or remind you of some overview on the modularity maximization with the example of a chromatin contact network, which was actually the main topic of my talk in this very workshop 2 years ago. And the latter or main part of my talk today is about systematic treatment of community reorganization inspired by the things we learned from such experience on real network. So let me start to talk about this series of work by introducing the modularity maximization. As many of you will already be familiar with it, a community structure of a network refers to this kind of a division of the entire network into pieces, where each of them is statistically more densely connected inside than the inter-community connections. This is a fairly intuitive concept, but at the same time, it's quite practical to see the structural and functional subunit lurking in a network system. And one of the most conventional ways to detect such a community structure is maximize this objective function called the modularity. So basically, it quantifies how many of the edges are intra-connected ones compared to the background or the expected one expressed by this null model term for a given community partition. And I'll come back to this null model part especially soon in the context of the chromatin interactions in a physical space. And here, the maximizing this function corresponds to finding the most densely intra-connected subpart. And a bit later technical but important part you should remember throughout my talk is this so-called resolution parameter gamma in front of this normal term. And it actually controls the overall scales of the communities bound, where the larger value of gamma tends to yield smaller communities or larger number of community as illustrated here. By the way, the another ingredient I will use later in this alternative formulation of the modularity function in terms of a group indices instead of the node indices. So I put this slide here for reference. And the derivation between them is actually surprisingly not available in literature, perhaps except for this wonderful textbook by Mark Newman, the Korean version of which is also available by the effort of the Korean network scientists, including myself. So first, to set the stage for my personal motivation to study community reorganization as a result of modularity maximization, I have to introduce this human chromosome structure. And chromosomes have this complicated, non-trivial folded structure inside the cell nuclei. And this project was actually initiated by this binational research corporation grant, so I'm thankful for it, and I'm still maintaining the collaboration with the Ludwig group in Umeå, Sweden. So when I first learned about this structure in biology class, I believe that all of the details about this packing structure are well known. But of course, that's not the case. So I think this is one of the many examples that are unknown, even if we decoded the human genome sequence itself. And deciphering this complicated structure is, of course, crucial to understand how this fundamental genetic material or chromatin works in cells. And a major breakthrough step to reveal the physical proximity between this genomic sequence is this interaction map of the chromatin loci called high-C. So this high-C technique provides the physical proximity mediating the interaction between the two chromatin loci in this matrix form. And you can see some non-trivial structures here and there, depending on the scale, which is in fact important clues to reveal the 3D structure. For example, there's this off-diagonal part in the higher resolution is the evidence of the loop structure, as the title of this cell paper suggests. Another famous example in lower resolution is this block substructure called topologically associated domain, or TAS, which obviously represent the local packing. So when I first saw this structure, I mean, as any of you would think, I realized that this is a very familiar problem to me, because if we consider the loci as the node and the weights between them as the weighted edges, detecting such a block diagonal substructure is, of course, precisely corresponds to the community detection problem in network science. And there is an important piece of information you can use here, as chromosomes are basically 3D folded structures from the linear sequence. In other words, we can actually clearly distinguish between the cases of the pairs of loci located far away and those very closely located in the genomic or linear sequence. And moreover, thanks to the study of this contact probability scaling as a function of sequence distance, we know that the background interaction between two loci follow the so-called fractal globule scaling, which implies that the contact probability is inversely proportional to the linear distance. So we use this fractal globule null model term composed of both the overall interaction for each node and this distance-based scaling behavior in the modularity function throughout this study. And take this generalized version of the famous Lubang algorithm for modularity maximization to detect such scale-dependent communities. In particular, I really want to emphasize the importance of using a proper null model term for community detection based on as much spatial information as possible known to us. Personally, I highly recommend this paper by Daniel Bassett and Mason and other colleagues on this problem, on this possible modification of the null model term from a more general perspective. Before we jump into a real high-C map data, we test our community detection algorithm with the various scales on the model structure called conformation dependent polymerization, or CDP fractal globule model, which supposedly mimic the chromatin packing with the fractal globule scaling. So with this example, I would like to emphasize the very important advantage of our method compared with the more conventional definition of TED, which is the ability to detect the non-contiguous communities like this segment here, A, B, C, and again, A and B. In addition, as the results for different resolution parameters are in principle or independent to each other, the communities across different scales are not necessarily strictly hierarchical, which is in fact a very important main topic throughout the presentation. And the best part of using this kind of model system is, of course, we actually know the detailed 3D structure. So let's see if our community detection scheme successfully find the complex structures of this model polymer system. And the plot on the left shows the scaling of the 3D distance versus the linear or sequence distance inside the detected community. And as you can clearly see here, the 3D or Euclidean end-to-end distance for a community boundary is systematically shorter than the random counterpart. And this clearly distinct scaling behavior for CDP models intra-community structure holds also for this rescaled 3D distance divided by the radius of the gyration as well. So it means that our community detection indeed successfully identifies loop or compact substructure. So based on this validation with this polymer model system, we move to the real high-C map data of chromatin interactions. So we use this particular high-C map data introduced in this famous Cell paper with the 100 kilobase pair resolution and converted the raw data to normalized version of the interactions with this particular normalization scheme. So, and by the way, this original Cell paper provides the set of the single resolution TED as some kind of a gold standard data, and we will compare it with our result later, of course. And here's the representative results of the communities we found with our method for the three different resolution parameters in the case of the chromosome 1. And here are examples of non-contiguous communities. I mean, these yellow pieces constitute the same community, and so these light blue pieces. And of course, we can detect the communities for every chromosome like this. And black stripes here represent the non-coding regions, by the way. As I already introduced, although many communities in different resolution looks like being nested, but it's not strictly true, as you can see from this magnified figure. And unlike the polymer model system, the real 3D structure of the chromatin interaction is not known, so we have to rely on some relevant biological factors for verification, of course. And the first signal we use is this well-known biological marker called CTCF, which is supposedly enriched near the chromatin domain boundaries. So we compare the CTCF enrichment with the community boundaries from our method with the text from the cell paper I mentioned as a kind of a gold standard. If there are significant overlap, it means that our method can detect the chromatin domain boundaries, just like the head in this cell paper. And of course, in contrast to single resolution TED, our method can detect the communities across various scales. So depending on the resolution parameter gamma, the overlap between community boundaries and the CTCF enrichment is changed like this. So as you can see here, for chromosome 1, around the gamma value near 0.7, the overlap between the community boundary point and both CTCF enrichment and the TED boundary points reported in the cell paper become maximum, measured by this precision value. If we adjust the resolution parameter properly, we can recover the TED with the property of the CTCF enrichment. So we successfully recover the TED structure at this particular range of gamma. Then what about the other ranges of gamma or resolution parameter? And one of the earliest findings about the chromatin interaction is the separation between so-called A and B compartment using the simple PCA, and one of which is more genetically active than the other. So if we detect the communities for very low resolution or low values of gamma, can you recover this A-B compartment separation as well? And it is of course possible in principle because these A-B compartments presented in this paper are non-contiguous checkerboard pattern, and our method can detect such a structure, which is something that cannot be detected with the many conventional test finding method, as I already emphasized. So here's the evidence of the recovery of A-B compartment from our method. So if we tune this parameter range low enough, then we actually obtain two communities. And as we can see from this overlap table, the largest community from our method roughly correspond to the B compartment, more inactive one, and the second largest community roughly corresponds to the more active A compartment. So if I summarize the results reported in this paper published 5 years ago, by tuning the resolution parameter in the community detection scheme, we could actually recover previously reported substructures of HiCMap. And based on this verification, we suggest a whole spectrum of community scale that may be meaningful for different biological contexts. And later, we have actually re-examined this semi-hierarchical structures of high-C community more deeply, which has eventually become a motivation to study the community reorganization problem in a more general context. So in this paper, published last year, we quantify how hierarchical or nested these communities are in different scale, inspired by this nestedness metric used in the ecological network studies. By setting up this mathematical formula to quantify from the most nested to the most segregated relation between the communities in different scale, we examine the level of hierarchy in this high-C community structure. So here, the perfect hierarchical structure would be composed of the community pairs, either fully segregated or fully nested structure, corresponding to these double peaks here in the rightmost and the leftmost part. On the other extreme case, randomly arranged communities would produce a certain distribution of this nestedness measure. So for our high-C communities, it's located somewhere in the middle, meaning that the chromosome communities are semi-hierarchical. So in other words, it's a mixture of hierarchical and non-hierarchical structure. And we successfully regenerated these various profiles with a simple hierarchical folding model starting from the smallest unit of our communities called domains. To elucidate the biological implication of this, we classify the communities into different chromatin states defined in this paper. So roughly speaking, here the regions marked from A to C represent the more active regions than the inactive heterochromatin or D region. So basically, our results show that the active regions are systematically more hierarchical than inactive regions. And all of these biological aspects aside, I think the important take-home message for us is the semi-hierarchical or reorganization of the community in a real network. So, collaboration with Won-hee and other previous collaborators on the topic of network community structure, I decided to work on this scale-dependent organization of the community detection. So basically, is this ideal picture of a monotonic community fragment as gamma increases? Is this the real life or is just fantasy? An anecdotal but powerful example is this striking plot, one he showed me about a year ago. At least it's really striking for me. For the archive condensed matter consortium network, it described the number of communities detected by the Lubang algorithm as a function of the resolution parameter gamma. And since I always had this simple fragmentation process in my mind as the resolution parameter gamma increases, when I first checked this plot, I honestly couldn't believe it. So I literally plotted myself and became convinced that it should be real. So again, I sincerely apologize for doubting my trustworthy postdoc results, but it's definitely worth further investigation. I mean, this phenomena. I mean, what's going on here? So how can the number of communities become decreases as gamma increases, somewhat paradoxically, which supposedly detect the smaller and the smaller communities? So to investigate this, I first asked my good old collaborators on network community structure about this phenomena. And that was just a simple question. Have any of you ever seen this kind of behavior? And it turns out that Dr. Daekyung Lee in the middle recall a weaker version of this in one of the data set I will introduce later. And most thankfully, they decided to join this project without any hesitation. So we were teamed up and started to seek a possible explanation. The first observation to get a clue is something, well, any statistical physicist would think, which is the size of the community, especially the dominant ones. The first thing we noticed is that this largest community size is actually monotonically decreasing as gamma increases, despite the ups and downs for number of communities. Meanwhile, the second largest community size, starting from zero, obviously by construction, of course, very abruptly grows at a suspiciously similar value of the starting point of this decrement of a number of communities here. And the most plausible scenario, therefore, would be the growth of the second largest community accompanying some sort of aggregation of the small communities, possibly by observing them. So we decided to look into the most elementary process of community division, or more generally, community reorganization, as our title suggests, in terms of the objective function to maximize, or the modularity, which is written in terms of the group indices, as I already introduced earlier, because it's a more convenient to present our argument here. So consider these two different community partitions hypothetically optimized for different gamma value, the gamma 1 and gamma 2. And the basic group merging criterion of this modularity maximization is expressed by sign of this change in modularity, which only involves the sum of the weight between the two groups and that attached to each group. And the resolution parameter gamma here controls the relative competition between these two terms. So if you look at this term, it's intuitively understandable that larger value of gamma would prefer smaller communities overall, but it's a bit more subtle as we will see in the next slide. So this hypothetical situation can be recast as four inequalities regarding the merging relation between these three groups. Under this condition, if we carefully examine the possibility that G3 is merged into G1 and G2 union at gamma 1, and possibility that G3 is merged into G1 at gamma 2 larger than gamma 1, this situation is possible in principle because this product of the gamma and Kg determine the sign of the modularity change. So in this case, the largest community size is decreased as gamma increases, while the number of communities is conserved as just the 2. So one can imagine the possibility of even the decreasing number of communities if we assume a bit more complicated scenario. So in other words, the number of communities is not necessarily monotonically increases as the gamma increases, considering the functional form of the modularity. Just in case you are really, really curious about the derivation of the delta Q formula, here's the slide for that. But as I expected, there's no time to go through every step. So please check our preprint for detail. So let's get back to more intuitive part, which is looking at the organization of communities of different sizes near the resolution of our interest. So here, each node on the right panels represent the community as a supernode, and the edges are connected if there's an edge between the nodes in each supernode. And initially, a single community started to be fragmented by giving up much smaller supernode to form this kind of a star network composed of supernode or superstar, according to my dear collaborator Hite. And as gamma increases further, with this coarse-grained picture of community, we can clearly see that our earlier assumption that this rapidly growing second largest community is kind of growing by absorbing smaller communities, which result in the decreasing number of communities as gamma increases. Perhaps the most intuitive illustration of this situation to the audience would be the simple but powerful model network coined by Won-hee, which is composed of the two big cliques here and small peripheral triangles. So for small gamma, these two cliques are stuck together as the largest community, while all the small triangles are separated community. So there are 7 communities in total. And if we increase the gamma a bit, the two cliques here are actually separated, but both of them reobserve all the small triangles attached to them. So the number of communities now become 2, suddenly decreased from 7. So we take a step further into individual node community membership in this archive collaboration network in this nice form of the Sankey diagram. And indeed, the initial fragmentation here of the single community into giant large community and the rest of the 61 communities, and reobserving process happening here, yielding 38 communities at the larger gamma values are clearly visible. So compared to the previous example, each of these resolution here would correspond to this and this. And another example is the World Airport Network, where this decreasing part here correspond to the big Atlantic division accompanied by the reobserving of a small Alaskan and Canadian airport by this American cluster. And a bit of a behind story here is that my collaborator on previous project, Daegyang, in fact found this part a few years ago, but at the time we didn't pay much attention. Perhaps because the level of decrement is not that prominent. Much less prominent than the archive network. And I'm really glad to have a framework to explain this after this obviously missed opportunity. Finally, by creating this simple SBM, we successfully recreate this nice plot showing the community reorganization observed in a real network. So our rule of thumb is that if a network has this type of heterogeneously organized structure, this community reorganization can be rather ubiquitous throughout many types of network. Of course, the non-trivial reorganization would be minimal for a strictly hierarchical structure with the relatively uniform pieces, but we can guess that such a case would be exception rather than norms. So here's the summary and some take-home message for you. And despite a couple of decades research in network communities, we still believe that there are things to careful re-examination, such as the scale-dependent reorganization I presented today. And our natural next step would be investigate more ultimate origins of the various types of a community organization. So thank you for listening, and please check our paper and preprint for details.